All right, this video is going to cover muscle contraction. We'll go through the phases that go on at the level of a muscle cell, right, the muscle fiber there, and then we'll go through contraction of whole muscles. So contraction involves, first of all, coupling the motor neuron excitation to the excitation of the muscle, and then spreading that throughout. That's phase 2 there, excitation, contraction, coupling. The actual movement of actin and myosin, the contraction, and the relaxation of those fibers. So that's shown here in this cartoon. So phase 1 there, that's going to be the excitation. A motor neuron excites the muscle cell, generating an action potential that spreads throughout. As that action potential goes into the T tubules, it activates voltage-gated calcium channels along that sarcoplasmic reticulum and release calcium. That is our excitation-contraction coupling. Once we release calcium, that then drives the next phase, contraction, where we decrease muscle length, right? We're going to slide those thin filaments along the thick filaments. So myosin here in the middle, the thick filaments, pulls the thin actin filaments toward the middle, and the muscle contracts. Relaxation occurs when we stop exciting the muscle cell, we stop generating action potentials, we stop releasing calcium, and so actin and myosin can no longer interact, and so those thin filaments are no longer pulled toward the middle, and the elastic elements pull them away, increasing muscle length, and that's relaxation. Okay, so for excitation, we're going from neuron action potential to muscle action potential. So the action potential, of course, depolarizes the presynaptic site. Calcium comes on in, and that leads to the fusion of presynaptic vesicles. There's 3 there, should be about 20 times more than that. So neuromuscular junctions are massive, and we release lots of acetylcholine. All right, now we're out of the motor neuron, we're into the synapse, and acetylcholine binds to acetylcholine receptors. And these, at the neuromuscular junction, are simple ligand-gated cation channels. Ligand-gated means they open up when acetylcholine binds. Cation channels means they're going to let positively charged ions move into the muscle, so they depolarize the muscle, generating a local potential called an end plate potential. Lots of sodium comes in, a little bit of potassium leaves, right? But the sodium overwhelms the current here, and so we get depolarization. All right, that then goes from ligand-gated ion channels, right? That depolarization that comes in, green means go here, so that not acetylcholine, but sodium, right? Positive charge comes in, and that then triggers the next ion channels in line, voltage-gated ion channels. Sodium depolarizes, potassium hyperpolarizes and repolarizes. This works exactly like any other action potential. So we hit threshold because of ligand-gated ion channels, and once we hit threshold, the voltage-gated ion channels then propagate the depolarization. That's this yellow arrow here, that's my action potential, along the surface. Now, of course, in muscles, we got those transverse tubules that bring the surface down deep into the muscle. And so now all of the muscle, both the surface and the inside of it now, are depolarizing. That depolarization then is going to spread into the sarcoplasmic reticulum. And of course, we remember this is where we have our store of intracellular calcium. So there's the terminal cisterns here on each side of the T tubule. This surrounds it in real life, but in this cross section, one side here, one side here. So the sarcoplasmic reticulum filled up with calcium, and voltage-gated calcium channels get turned on by the action potential in the T tubule. So the sarcoplasmic reticulum then spits out calcium. Calcium binds to this protein called troponin on the thin filaments. When troponin binds calcium, it changes shape and pulls this second protein, tropomyosin, out of the way, exposing the myosin binding sites on actin. And there's my excitation-contraction coupling. I go from an action potential in the muscle to now exposure of those myosin binding sites on the thin filaments. So here it is in cartoon format. I've got my yellow thin filament here. Some calcium is floating around, right, because I fired an action potential. Calcium binds to troponin on the thin filament. That then moves tropomyosin out of the way, just like in this cartoon. There we are. So now my myosin binding sites are exposed on the thin filament, and myosin on this thick filament here binds. And there I've formed my cross bridge, and I'm ready to start the third phase, which is contraction. Here's where myosin is going to increase tension on those thin filaments, pulling on them and decreasing the overall length of the sarcomere. So this would be the sliding filament theory. And for this to make sense, you have to understand the myosin binding preference. Myosin can only bind one thing. It's either binding some adenine nucleotide like ATP, its favorite thing, or ADP, its least favorite thing. It also has an actin binding site, so it can bind actin as well. And notice its preference. It wants ATP, it'll take actin if it can't get that, and ADP is its least favorite thing. Now, myosin is an ATPase, which means that after it binds ATP, it converts it to ADP. So it takes its favorite thing and makes it its least favorite thing. And that's where our story begins. Myosin is, of course, holding on to ADP because it's an ATPase. So it breaks it down to ADP. Now it's bound to actin, forming the cross bridge. Hey, it can only hold on to one thing, and which does it prefer? Actin. So it's going to let go of ADP. And when it does that, it changes its shape. And it goes through the power stroke where it bends by about 90 degrees. That's what's happened here. So the myosin head has cocked back, pulling the actin filament, 10 nanometers, in what's called the power stroke. Okay, so we're holding on to actin, and ADP has gotten out of the way. Now my adenine nucleotide binding site is open, so I can bind ATP. I can only hold one thing, so when I bind ATP, I have to let go of actin. So ATP binding breaks the cross bridge. But I'm myosin. I'm an ATPase. So I hug it, I squeeze it, and I convert ATP back to ADP. Now here's the fun thing that happens when I do that. I cock forward. So the shape of myosin depends on whether or not it's holding on to ADP. If it's holding on to ADP, it is cocked forward in what we call the recovery stroke. When it lets go of ADP, it cocks back in what's called the power stroke. ATP binding breaks the cross bridge. And the release of calcium there in our excitation-contraction coupling allows the formation of cross bridges. When cross bridges form, I spit out ADP. So let's see that in a cycle here. So here's the cartoon, right? We break ATP down to ADP. That's where we're at right now, cocked forward. So we've had the recovery stroke. Because calcium's there, the binding site is open, I form the cross bridge. That's what happens over here in step 3. I then release, well, there goes the phosphate. That's to stabilize the interaction. That then causes me to release ADP because I can only hold one thing. I then cock back 10 nanometers. That's the power stroke. Hey, my adenine nucleotide binding site is open, so now I can bind ATP. That's going to happen over here. Nice little green ATP molecule floats in. Great. I can only hold on to one thing. I prefer the ATP. So we break the cross bridge there. Now I'm not holding on to the thin filament. Of course, I'm an ATPase. So I'm going to break that ATP down to ADP and a phosphate, leading to, of course, the recovery stroke. And this is going to happen over and over and over and over asynchronously in a whole bunch of these little myosin heads here on the thick filament. And that's going to pull my red thin filaments here toward the middle, going from a relaxed to a contracted state. So the sarcomere shortens. There they are right there, shortening. There's our movie here, and we'll watch the sliding filaments here take place. Myosin on the thick filaments. You'll notice we're going to have a whole bunch of asynchronous contractions. Calcium's floating around. It's binding to troponin, moving tropomyosin out of the way in the thin filaments. We're now pulling the thin filaments toward the middle, and there you have it. Now, they're not all working together. This is not a rowing team. They're asynchronous. So each individual myosin head is doing something slightly different from its neighbors. About half of them are attached via cross bridges, going through the power stroke. About half of them are detached, no cross bridge, and they're going through the recovery stroke. So this keeps tension on the thin filaments and prevents them from slipping back, right, and then lengthening again. It also makes sure we don't have jerky muscle contractions. So not all the myosin's pulling. So there's about an equal, steady tension on those thin filaments. And so we get nice, smooth muscle contraction without slippage. That contraction shortens the sarcomeres by up to, as you can see there, the magic number 40% during any given contraction. By changing the length of all those many sarcomeres, those serial sarcomeres, the whole muscle changes its length, as we can see here, right? If we look at the distance between these Z discs, relaxed, contracted, they're a little bit closer. Okay, so how do we relax? Well, stop firing action potentials in the motor neuron. What that's going to do is decrease acetylcholine release. So no more acetylcholine release. That acetylcholine, when it unbinds the receptor spontaneously, gets broken down by cholinesterase. So no more acetylcholine in the synapse. No more depolarization of the muscle. No more action potentials. No more depolarization of those voltage-gated calcium channels, so no more calcium release. Instead, I just get this continual reabsorption of calcium via calcium pumps. And steps 15 and 16, these are always going on, by the way. I'm always degrading acetylcholine, and I'm always cleaning up calcium. However, this doesn't make a noticeable dent in the amount of acetylcholine or calcium unless my motor neuron rests. So I have to stop making a mess so that this slow, continuous cleanup job finally makes a dent. Once I've cleaned up most of the calcium, then it's not bound to troponin anymore, so troponin doesn't pull tropomyosin out of the way. So now the myosin binding site is covered again, no more interactions. So this myosin head here is going to hydrolyze that ATP, do the recovery stroke, and wait for the next burst of calcium to move tropomyosin out of the way. Now, muscles are going to rest somewhere around this kind of optimal resting length for their sarcomeres. You can see the rough ballpark there, just a slight bit over 2 microns for those of you keeping score at home. If it gets much shorter than that, or much longer than that, the amount of tension that it can generate decreases. Notice we have this kind of bell, not really a bell, more of an inverted U-shaped curve here for sarcomere length, that's on the X, and the amount of tension that can be generated, that is the strength of the muscle. So we want to have some overlap so we can have our thick filaments pulling the thin filaments. If we don't have any overlap because we're overly stretched, well, we can't generate any tension. Or if we're completely contracted and my thick filaments are already smashing against that Z band there, no more tension there either. I can't contract anymore. So we're resting somewhere about in the middle. The reason that we rest there is about twofold. First of all, the anatomical limitations of joints prevent muscles from being overly stretched to this tension-free length here. Secondly, our muscles and our motor neurons are never really silent in the long run. There's always some degree of activity, what we call tone, muscle tone, motor neuron tone. They're always a little bit active even at rest. And that, along with our anatomy, keeps our muscles resting at a length that makes them able to generate optimal force. Okay, none of this is free, by the way. So muscles, just like nervous tissue, are quite expensive, so they burn through a lot of ATP, and there's 3 things that ATP are doing for us. Of course, the sodium-potassium pump still exists in muscle cells, so we're spending an ATP to move out 3 sodium and move into potassium, and this makes sure that I have the correct ionic imbalance so that I rest at about, I don't know, minus 80, minus 90, something like that. Muscles are a little more hyperpolarized than neurons. So in order for me to rest, I have to spend ATP on the sodium-potassium pump, maintaining those ionic gradients that give me my membrane potential. And then calcium. I got to clean up that calcium. That ain't free. Remember, I'm always fueling those calcium pumps, and those could be semi-efficient, moving 2 calcium ions, or hell, maybe they only move the 1. I got calcium pumps on the surface to get rid of calcium, and I got a whole bunch of calcium pumps on the smooth endoplasmic reticulum, or the sarcoplasmic reticulum if we're in muscle. So SERCA here, smooth endoplasmic reticulum, calcium ATPase. I burn an ATP, I move calcium into the smooth endoplasmic reticulum, called the sarcoplasmic reticulum in muscles. And there a protein called calsequestrin, name makes sense, binds to calcium and holds it within that sarcoplasmic reticulum. Okay, so ATP is there so I can rest and I can clean up calcium, and it's also there for the whole cross-bridge cycling that we just went through. ATP, when it binds to myosin here, breaks the cross-bridge. I then hydrolyze ATP for the recovery stroke. So I'm using ATP for many different effects in muscles, and the lack of ATP explains why rigor mortis occurs after death. So on the order of hours here, post-death, the muscle begins to stiffen in a process called rigor mortis. Okay, what's going on here? We're dead. No more blood movement, so we run out of oxygen. We're running out of glucose. We're not able to make ATP. So the first thing we lose in these 2 muscle samples here, so these are 2 different samples from rabbits, I believe. Time 0, we just died. We still got the normal amount of creatine phosphate, ATP, and my muscles are relaxed. Okay, whether you're on the top or the bottom, doesn't matter. The first thing to go is creatine phosphate. That makes sense. So we run out of this. Once we run out of creatine phosphate, can't make any more ATP, so that starts to decline, and here's where things start to get interesting. When I run out of ATP, what happens? Well, I can't maintain my membrane potential. That then leads to an action potential in my muscles. They start twitching. That, of course, leads to calcium release. That, along with reduced clearance of calcium, means that I have a whole lot more calcium floating around in my muscle cells. As ATP drops, more calcium. That explains why my muscle contraction curve here, the filled circles, increases as ATP levels drop. So I go through the cross-bridge cycle, I contract my muscle, I burn up even more ATP. And then when I essentially run out of ATP here, my muscles stay contracted. And that's because of this third bullet point here, I can't break the cross-bridge anymore. So part of it has to do with we're not moving blood around. Part of it's just, muscle is kind of rotting and degrading. And so as the sarcoplasmic reticulum degrades, calcium gets released uncontrollably. That's the early phase here. After a few days, rigor mortis goes away, and that's because the myofibrils have started to rot. So as the actin and myosin break down, there's nothing holding that muscle tension anymore because, the muscle is degrading, and the rigor mortis goes away. All right, so there we have contraction in a muscle fiber. You can scan this QR code if you want, or you can follow the link in the description, and make sure you got something out of that. Okay, now we'll go into contraction of whole muscles here, and the whole point is to generate some tension. If we generate more tension than there is load on the muscle shortens. If we generate less tension than there is load on the muscle lengthens. Just that simple. Now, the contraction and relaxation of a muscle is what we call a muscle twitch, and they are variable amounts of time depending on if it's fast twitch, slow twitch. You get the point here. But regardless of the actual numbers, they all follow the same basic format. We get some stimulation and there's a latent period. Part of this has to do with, of course, exciting the muscle and spreading the action potential and spitting out calcium. And then there's also removing the tension of the elastic components on the sarcomeres. So there's a little bit of a delay between motor neuron input and the actual generation of muscle tension, the contraction. But once we remove all that tension and we start to, I'll actually remove the elastic components there. Once we put tension on them, we now contract, and that's my contraction phase. Now I'm generating external tension, right? I'm pulling the Z discs closer to the middle. This requires continuous release of calcium, right? So I keep those actin-myosin interactions taking place. Of course, also requires continuous presence of ATP. Relaxation occurs after that. If I stop releasing calcium, I will relax, and it will take longer than contraction. It's a variable amount of time, but that's what's going on here. I'm cleaning up the calcium. I'm decreasing actin-myosin interactions, and that's the relaxation phase. Now, the strength of a contraction is going to depend on the strength of contraction within a single fiber and the whole muscle. Okay, the fiber, we've seen the length-tension relationship. So that's going to determine how strong the contraction is. If it's already contracted, hard to generate more force. Fatigue matters, and we'll cover this in our next lecture together. For those of you who won't be there, fatigue is largely a consequence of the accumulation of potassium and ADP and phosphate. And what those do is weaken the muscle's ability to fire action potentials and go through the power and recovery stroke in myosin. Temperature matters. So a warm muscle will indeed be stronger, and that's because myosin is an enzyme, and the lower the temperature, the slower the enzymatic reaction rate. So a warm muscle will hydrolyze ATP faster and go through the power and recovery strokes faster. Hydration matters. You need to have enough water in there to make sure your actin and myosin are, optimally spaced. This is not something you need to worry about. This is something that's going to be through like extreme dehydration. So sip your water. No need to worry much about hydration. As long as you don't feel thirsty, you're probably fine. Stimulus frequency matters, and we're going to see this more in a little bit, so just put a pin in that for now. But the more frequent a motor neuron depolarizes the muscle, the more tension it will generate. Now, the muscle itself, of course, depends on the strength of all of its fibers, so all these bullet points up here are true for the muscle. And on top of that, we have to ask ourselves, how many motor units have we recruited? And that depends on the intensity of the stimulus, as opposed to the frequency here. Okay, so a motor unit is a motor neuron and all the muscles that it innervates. And what we're doing up here, we're delivering some voltage, levels 1 through 9, meaning more voltage. So we're shocking a nerve, and these subthreshold shocks don't turn on any of my motor units. So none of my axons are orange here. They're all silent. Once I hit a stimulus of about 3 here, okay, that's enough to turn on one of these motor neurons right there. And so I get a little bit of muscle tension because this has a relatively small motor unit. As I deliver stronger shocks to the nerve, I turn on more motor units and I get stronger muscle contraction until I've turned on all the motor units. Doesn't matter if I shock that nerve any harder. I've already activated all 7 of these motor units, and so I get the same maximum contraction. Okay, so let's dive into that tension-frequency relationship that we just went through. So when a muscle fiber twitches, if we allow it to fully go through the relaxation phase before we trigger the next twitch, we're going to get really no overall change in muscle tension. Yeah, we get a little contraction, relaxes, but it's the same. This peak isn't moving anywhere. In order for us to generate tension, I need a higher frequency stimulus here. So let's go over here, high frequency stimulus. I contract, I relax a little bit, but before I get all the way to baseline, I'm contracting again. And so I'm building on top of the previous twitches here, allowing me to build up more tension in the muscle. And this would continue on over here, and I would hold what's called incomplete tetanus, the fluttering maximum contraction of muscles. Now, of course, when a muscle contracts, it's not waving around like this. This is for an individual muscle fiber. Just like we have asynchronous contraction of those myosin heads within a muscle fiber, each muscle fiber contracts asynchronously. So while one is contracting, the other's relaxing. While this one's relaxing, its neighbors are contracting. And so these jerky movements of individual muscle fibers cancel out, and I get smooth contraction of the entire muscle, right, of the motor unit itself. So the muscle will increase tension smoothly, but every individual muscle fiber is going to go through these twitches. Now the tetanus that we hold should, again, oscillate like this. That would be incomplete tetanus. This is if you're in the gym, right? You're contracting your muscle there, and you're trying to lift, and it's not working, and you're just kind of fluttering back and forth. This is fine. This is what then makes the muscle grow. What we don't want to see is this flat line, this complete tetanus. This is where the muscle has maximally contracted. You won't see this. You'll only get this in the laboratory, and this is always damaging to muscles. Luckily, you don't have to worry about that. I'm sure you work out hard, but you're not working out that hard. Okay, so those motor units that we turn on, we're not turning on all of our motor units at the same time, right? We're going to start with only the minimal and work our way up. So we have this thing called the size principle. So upper motor neurons are going to recruit the lower motor neurons that create those motor units, starting with the smallest and then working their way up to the larger ones. Now, the reason that this works is because the lower motor neurons, as we can see down here, in this cartoon, vary in size. Some are smaller, and they relate to smaller motor units. Probably because they're getting less neurotrophic support, and so they don't grow to be as big. But why does that matter? A smaller neuron has higher resistance. And if we look at Ohm's law here, if I'm delivering the same current to all four of these neurons, who gets the bigger potential? This one right down here, because it has a higher resistance. So if I have higher resistance times the same current as my neighbor, I'm going to have greater depolarization. So the first ones to get excited are the small motor units, the ones that generate relatively little force. And so I'm not going to get any sort of herky, jerky muscle movements. Instead, I'm going to turn on the weakest motor units. If that's not enough, my upper motor neuron works a little bit harder. I strain a little bit harder, and I turn on the next motor unit. If that's not enough, I try a little harder, and I turn on the next motor unit. And if I need to do something really strong, then I turn on all of my motor units. My upper motor neuron is maximally active. You're really straining. You're trying to get a thing done. We're delivering as much current as we can to finally turn on these large motor units, and we deliver, boom, maximum contraction. And look at that. It's, of course, not a flat line. We get that incomplete tetanus. And that can be with a whole lot of force because I'm including my really large and strong motor units, and the incomplete tetanus might be down here just because I only turn on the small motor units, and that's all the force they can give me, but they can give me that force all day. So the size principle here is a wonderful thing. It's explained by Ohm's law, and what it provides for us is that gradual recruitment from weaker, smaller, to stronger, larger motor units. And this makes the muscle far more efficient. These larger motor units are more expensive. They're going to burn a lot more ATP than the smaller motor units. So why use them if you don't need them? So then we start our muscle contraction, right? We've turned on the motor unit, whether it be large or small, doesn't matter. What we're first going to do is increase that muscle tension, so the tension within the muscle. This would be the isometric phase, so we're not changing muscle length here. What we're doing is getting rid of those, elastic elements within the muscle. We're increasing that internal muscle tension. Then we enter the isotonic phase. Once the muscle starts to shorten, that's enough. No need to put on any more tension than that. We'll hold we'll hold the tension there, right? So isotonic, same muscle tone. So let's not recruit any more motor units. Keep that number, that got the job done, and the muscle length decreases. So during this phase, we're taking out the slack in our muscles and we're recruiting motor units until we have enough of them to change muscle length. This is to make sure that we're not wasting energy by getting in those larger motor units unless we need them. Then we can use those same terms and talk about muscle contractions. So in an isometric contraction, you're not changing muscle length. Iso, same, metric, distance. And this is the beginning of any muscle contraction. So there's always an isometric phase. Now, we might actually not ever go beyond that. We might keep the contraction isometric in order to stabilize a joint or just maintain posture, maintain the length of a muscle. We don't really want to move the body. We're just in that isometric contraction, maintaining muscle tone. Okay, isotonic contractions are used anytime we want to change muscle length. So sure, it might start off isometric, but we actually want to lift. We want to move this dumbbell around, and that movement can be in one of two ways. It can be the concentric contraction there, where the muscle shortens. So now we're isotonic. We're going to keep the tone the same, and it's enough tone to overcome the load and move the dumbbell upward. So we shorten the muscle. We could also have an isotonic eccentric contraction, where we maintain tone, but we're maintaining it below the level needed to move the load, so the muscle lengthens. Now, in any movement, we're kind of doing both of these things. If we load heavy weight on here and we're trying to do the negatives, right, we're getting those isotonic eccentric contractions. These are a little more likely to lead to injury because, well, we're overloading the muscle kind of by definition, right? And so these are both important. Just be, a little careful here. Don't hurt yourself. And don't hurt yourself on these either. They shouldn't be painful. You should know everything you need to answer these questions. You can scan this QR code. You can follow the link in the description. If anything seems strange, shoot me an email. If you can't figure out how to do that, I think you can comment on this video, and I'll probably hear or read your comment and leave some words of wisdom if I have any. All right, I'll see you in the next one.